This activity allows us to explore how Agrobacterium tumefaciens, known as the nature's genetic engineer, transfer genes into plant cells. Through modeling and simulation, we visualize this process safely and creatively, without using real bacteria. Let's go through each phase. In the first phase, we focus on the T plasmid, short for tumor-inducing plasmid. This special circular DNA inside Agrobacterium is what enables the bacterium to transfer genes to plants. We constructed our own D plasmid model using colored paper. We used the red paper for the tDNA region, containing tumor-causing genes. We used the blue paper for the virulence region, which helps transfer DNA. We used the green paper for the upon catabolism region, which lets bacteria use plant-made nutrients. The last paper is yellow for the origin of replication, which allows the plasmid to make copies of itself. We also added border sequence that mark where tDNA begins and ends. These steps help us understand that scientists disarm the plasmid by removing harmful genes and replacing them with useful ones. For example, genes that make plants resistant to pests or drought. Next, we move on the actual simulation of transformation. A large container as the plant cell, a smaller container as the bacteria cell, and the beads to represent plant chromosome and proteins. Inside the plant cell, we placed 10 beads of one color to represent chromosomes, and the bacterial cell contained our T plasmid model. The containers were positioned about 12 inches apart. Here, we simulated plant injury by drawing plant signal cards. The signals we drew were sugar molecules and phenolic compounds. Both are natural plant wound signals. When these signals were detected, the Agrobacterium responded by moving closer to the plant cell. This represents how the bacterium senses and it's attracted to wounded plant tissues in real life. These signals also activate the Vir genes inside the bacterium, preparing it for DNA transfer. After signal detection, we simulated the next stage. Stage A: Attachment and Vir gene activation. We use colored beads to show proteins being produced. Blue beads for Vir D proteins, responsible for DNA cutting and coating. And the red beads for Vir B proteins, responsible for DNA transfer machinery. Stage B: tDNA processing. We cut out the tDNA region from our plasmid and attach a blue bead, symbolizing the Vir D2 protein that protects and guides the tDNA. Stage C: DNA transfer. Using our roll papers as a transfer channel, we move the tDNA complex from the bacterial cell into the plant cell. Stage D: Integration. Finally, we placed the tDNA among the plant chromosome beads and connected them to show successful integration, meaning the plant's genome now carries a new gene. Once the transformation was complete, we explored its real-life uses through gene engineering challenge cards. We were given examples such as creating golden rice enriched with vitamin A, developing herbicide-resistant soybeans, and producing plants that can manufacture medicines. For each scenario, we identified which beneficial genes could replace the tumor-causing ones, what selection markers would be needed, and what benefits and potential risk might occur. We also compared Agrobacterium-mediated transformation with other techniques like gene gun, electroporation, and viral vectors, and learned that Agrobacterium is one of the most efficient and natural methods for gene transfer. To conclude, these laboratory simulations help us understand how Agrobacterium tumefaciens acts as a natural genetic engineer. We experienced how gene transfer occurs from signal detections to DNA cutting to integration inside the plant genome. Our result, where sugar molecules and phenolic compounds activated transformation, clearly showed how plant wound trigger bacterium's response. For this fun and creative experiment, we realized how genetic transformation contributes to producing crops with higher yields, better nutrition, and greater resilience, truly shaping the future of agriculture. Thank you and good day.